DNA is the molecule that stores all genetic instructions needed to build, maintain, and operate a living organism. DNA consists of two complementary strands. Adenine pairs with thymine, while cytosine pairs with guanine. Before a cell divides, it must create an identical copy of its DNA through replication. Replication begins at special DNA sequences called origins of replication. The enzyme Helicase unwinds the DNA and breaks hydrogen bonds between base pairs.
As DNA opens, a Y-shaped structure called the replication fork is created. Single-strand binding proteins stabilize the separated DNA strands. Primase creates short RNA primers that provide a starting point for DNA synthesis.
DNA polymerase starts building a new DNA strand by matching complementary bases.
The leading strand is synthesized continuously in the same direction as fork movement. The lagging strand is built in short segments called Okazaki fragments. DNA ligase joins the fragments together, creating a continuous DNA strand. Replication produces two identical DNA molecules, each containing one old and one new strand. Next, the cell copies genetic information from DNA into messenger RNA, or mRNA. RNA polymerase recognizes a promoter and attaches to the DNA.
The enzyme opens a small section of DNA to expose the template strand.
RNA polymerase builds an mRNA molecule using the DNA template sequence.
When a termination signal is reached, the completed mRNA is released.
In eukaryotic cells, mRNA is processed before leaving the nucleus.
The mature mRNA exits the nucleus and enters the cytoplasm.
Translation begins when a ribosome attaches to the mRNA molecule. The ribosome reads mRNA in groups of 3 nucleotides called codons. Transfer RNA, or tRNA, delivers the correct amino acids for each codon. As amino acids join together, a protein forms and folds into its functional shape. This is how genetic information, DNA stores information, transcription copies it into mRNA, and translation turns it into protein. Like and subscribe for more biology animations.